Hi everyone, good day. So for this session, we are going to discuss what are proteins and what are the monomers of proteins. So let's start. So this is our objective for this session. First is to explain the protein structure, the functions, the synthesis, the processes, the levels of organization, and its clinical significance, which is, of course, these are the objectives in our SEMSTL, but in this case, I no longer tackle the clinical significance. And then the second one is explain what are the types of protein structure. Namely, we have the primary, the secondary, the tertiary, and the quaternary structure of the proteins. However, my PowerPoint presentation is quite long, so I'm going to cut it into two video presentation. So for this video presentation, we will only discuss the first objective. And then for the second video presentation, we are going to discuss the protein structure. Okay. So without further ado, let's start now our discussion. Now, let me ask you this question while listening to my video discussion right now. Why proteins are important? What do you think? Why proteins are important to us, not only to us human beings, but as well as with the other living organisms. Now, for human being, or for us, human being, as an example, as representative of living organism, proteins are very important simply because it serves a crucial in varieties of purposes and importance in order for us to survive. And you might be familiar of all of these things, and these are all made up of proteins. The muscles that you have in your body, all of your muscles, were made up of more than 600 muscles in our body, made up of protein. We have our hair is made up of protein. Our nails are made up of protein, and we also all have our skin as one of the one of the part of our body, which is also made up of protein and many more. So later on, we will find out why protein is important. Also, in micro levels, these are some of the examples in which they are made up of proteins. The antibodies in our body as part of our immune system that ward off and fight pathogens, those foreign materials that invade in our body, these are made up of proteins. Our enzymes for increasing our responsible for the metabolism in our body. This is also made up of protein, as well as the transport protein in the plasma membrane in cellular level still, in which it helps transport the molecules from the outside environment of the cell to the inside of the cell for proper cell functioning. Okay, so these are some of the examples in which they are made up of protein. Now, let's start with the characteristics of proteins. What are the characters of the proteins? Now, protein is being defined as a long chain molecules which is made up of strings of amino acids joined together. Okay, again, so this is a long chain molecule of strings of amino acids joined together. Since protein is an example of a polymer, and this is a polymer of amino acids, and amino acids are monomer of protein. So if we're going to synthesize the protein, it will form into amino acids, and at the same time amino acids, if we're going to what um anabolism, it will form into a protein. So all amino acids are compounds with similar basic structures, and these are their structures. But anyhow, let me show you this picture presentation. So this is a protein in which this is made up of long chain of molecule strings of amino acids. These are some examples of amino acids. Different amino acids, different colors represents different amino acids. The same for green, the same for violet, the same for pink, the same for yellow one. So these together, join together by a hydrogen bond in their backbone to come up with protein. And specific amino acids are made up of these three significant parts. We have the amino group, which is attached in the carbon molecule. We have the carboxyl group attached here also in the carbon, as well as in the side chain or the R group. So most likely in the amino acids, they are all of the attachment. Say we have the amino group, the carboxyl group, and the side chain will fall in their alpha carbon. Now, talking about the side chain or the R group, this is responsible for the varieties of amino acids. So because of the differences in the side chain or the R group attachment, come up with a different types of amino acids. Okay, so this is an example of space filling diagram for a representation of say we have what specific amino acid. So here we have this one, the attachment of all of these things in the alpha carbon. We have the carboxyl group or the carboxylic acid group. We have the amino group, and then we also have the side chain. Now again, as I've mentioned earlier, take note that side chains determine the amino acid's properties, and this is the only part that varies from amino acid to amino acid. Not their carboxyl group, not their amino group, but their side chain attachment. One specific amino acid have one specific side chain attachment. Now, further in addition, we also have what with regards to or in relation the context of the definition of protein, we have this general and specific definition. So the one that I presented earlier encompasses the general definition of what protein is all about. And again, protein is a monomer units. The monomer units of protein are amino acids, since proteins is an example of polymers. Same goes with the carbohydrates that serves as the polymer, and then the monomer of carbohydrate is what what they call that one, glucose. And then for lipids, that's a polymer. The monomer is triglycerides, and what the glycerol, yeah, the triglycerides and the glycerol and the fatty acids. And then we also have this one, the specific definition of the protein. So this is a peptide with at least 40 amino acid residues which are present. Again, for a specific definition of protein, they have this at least 40. That's the minimum amino acid, the chain of amino acid. So most likely there are already several proteins with higher than or more than 10,000 amino acids residues are known. They are named already. But common amino acids are only in between 400 to 500 amino acid residues. And for the small proteins, they only consist of 40 to 100 amino acid residues. Amino acid or the residues itself represents the amino acids. Now, more than one polypeptide chain may be present in a protein, in which they are classified into two according to their polypeptide chain attachment. We have this monomeric, which contains one polypeptide chain, and multimeric, which contains two or more polypeptide chain. By the way, do not be confused with the peptide and the protein. Peptide, these are short chain of amino acids. And when we say protein, these are the attachment of two or many, multiple, a lot of peptide bonds. If we're going to compare their sizes, peptide is smaller comparison to protein. Okay. Again, if one polypeptide is only what they call it, can only be found in specific protein, then that is what we know as monomeric. For multiple, two or more than that, that's multimeric protein. Now, protein variations are caused by the following reasons. There are three most common reasons why there are variations, why there are many proteins, why there are different proteins. So number one, we have the order of amino acids. So say for instance here in this picture, we have it here. This is the order of amino acids, and one detachment of amino acid already give another protein, or interchange of amino acid from this one to here already give another protein. And then we have the chain length, of course, smaller or a larger chain. And then the third one, we have the folding of the chain here, this one, in which it gives their distinct ways. And most likely in the future, in my second video presentation, we will discuss more about this one, since the folding of the proteins, giving it more giving more 3D or three-dimensional appearance will greatly help them serve their functions or the purpose of specific protein. So in this case, for the folding of the chain, I'm sorry for that, the one which is responsible for this one is the backbone of amino acids, for the chain of amino acids. So the most abundant molecules in the cells after the water, which accounts 15% of the cell's overall mass, is the protein. That's why this is also protein is very important. We need to consume protein in our diet. And the elemental composition of protein is made up of carbon, hydrogen, nitrogen, oxygen, and sulfur. So again, this is an example of a space filling diagram. So we have the gray one, which represents the carbon, the red one, the oxygen, the blue one, the nitrogen, the white one, we have the hydrogen, and the yellow one, which represents the sulfur. These are the composition of the proteins. And many specialized proteins, let's say for instance, they would serve their purpose according to their purpose. So they have a lot, or there are other elements which is being attached to it. So for instance, we have the ferritin. They have a lot of iron inside this protein. So we have iron, phosphorus, and other metals which is incorporated in the structure of specific protein. That is why they are known as a specialized protein. And with regard to the protein diet, we have here the representation of the percentage. I hope you can read this one, because I think this is what you cannot really read this one. The yellow one represents the carbohydrate. We have the vitamins and minerals, the protein, and the fat. 14% of our diet came from, or should came from, the protein, and most likely for our diet. So where can we found, or where can we get proteins? So we have meat, we have egg, we have cheese, nuts, fishes, and yeah, the beans, fish, and nuts. And when we consume protein, we the protein alone from the food that we eat cannot be directly be absorbed in our bloodstream so that it can serve its purpose. So the same as the discussion in our lipid metabolism, all the fatty food that we are consuming in our body, they cannot be transported inside our body to be used. So it needs to be metabolized. It needs to be metabolized, degrade further. So the same concept in the protein itself. So during protein metabolism or protein digestion, say we have the protein here inside the food, the bloods, it cannot be easily been up. It cannot be easily what can be uptake in our bloodstream. So therefore, it should be degraded in smaller portions, forming it into amino acids. Now, once already in the bloodstream, they will aggregate again to form the protein. So the purpose of detaching the chain of amino acid is just simply to pass through the bloodstream. Once they are already in the bloodstream, they will aggregate to form a protein in which they will serve their purpose inside the body. They will go in different pathways according to their functions and their purpose. And since we are discussing about amino acids, again, amino acids, these are the monomers of proteins. These are the building blocks of proteins. So let's try to get to know more what are these amino acids. So amino acids, standard amino acids, more or less, it varies. It depende sa references. Other references claim 20 standard amino acids, where other claim 21 standard amino acids. And these are some of the examples of those standard amino acids along with their representations and abbreviations, how they are being written. So let's say, for instance, we have alanine in the three letter representation for nomenclature represented by ALA. And then in one letter, we have represented by A, leucine, LEU, and then L, phenylalanine, PHE, then F, threonine, we have THR, and then T, cysteine, we have CYS, and then C, and so on and so forth. So here, a space filling diagram is also shown for you to further visualize their composition. Okay, say for instance, this one, the yellow one that I showed you a while ago for the elemental composition of the protein, specifically in their amino acids. So yellow represents the sulfur, right? The gray represents the carbon. We have white represents the hydrogen, and so on and so forth. And in addition, this is also another representation of the standard amino acid, the 21 amino acid, according to their classification. So amino acids can be classified according to their characteristics. So we have amino acids with electrically charged side chain, so either positive charge or negative charges. And these are some of the examples. We have arginine, histidine, and lysine for the positive charge amino acids. For negative charge, we have aspartic acid and glutamic acid. For polar, which have uncharged side chains, they are polar, however, they are neutral, and I will show you the definition for that one later on. We have the serine, the threonine, the asparagine, and the glutamine. And then we also have this one, amino acids with hydrophobic side chains. Hydrophobic meaning to say they are water-faring, because polars are hydrophilic, they are water-loving. So these are some of the examples for amino acids in which it belongs to this category. We have the alanine, the valine, the isoleucine, the leucine, methionine, phenylalanine, tyrosine, and tryptophan. Now for letter C, we have these special cases simply because all of these three, we have the cysteine, the selenocysteine, the glycine, and the proline, have different characteristics on their own that somehow do not fit to the category or the classifications of the three. Okay, we have the polar, the non-polar, and then the hydrophobic. So next, I'm sorry. So next we have now, this is one of my discussion a while ago. Hydrophobic amino acids, what makes them hydrophobic, what makes them hydrophilic, and then what makes them charged amino acids. Now, hydrophobic amino acids, they are hydrophobic simply because they are carbon-rich side chains, and there's no reason for other amino acid to attach in the water molecule to attach to them. For hydrophilic amino acids, they interact well with water simply because they can form a hydrogen bond into it in comparison to those hydrophobic amino acids. And for those charged amino acids, they interact with their oppositely charged amino acids or other molecules. So opposite meaning to say opposite charges attract. Of course, we know this concept already in our physics subject. And same charges repel. So positive will attract with negative, and negative will attract with positive. Positive, positive, no reaction. Negative, negative, no reaction. So same goes with the concept of the charges for the amino acids. The next one, we have now, in the context of amino acids, we have the derived and we have these essential amino acids. Okay. Now, for derived amino acids, these are non-standard amino acids. For the word itself, derived, they are the derivatives. So we have this specific other amino acids and there's something into it, so forming into another amino acids. Okay, the derivatives derive amino acids. So this is usually formed by an enzyme facilitated reaction in a common amino acid. After that amino acid has been incorporated with a protein structure. So I mean to say, this amino acid is being formed once one specific amino acid is being incorporated in a protein structure. Okay, so some of the examples are the cysteine, the desmosine, the isodesmosine, proteins or amino acids which are found in the elastin. Elastin, these are the proteins in our body which is capable of stretching. They are capable of stretching and then going back. So let's say for instance, we have our lungs, right? Our lungs are flexible. So we have expanding and then going back to its normal size. And then we also have this hydroxyproline and hydroxylysine, which is found in our collagen. So collagen is an example of protein, which is very important since this serves as a structural protein in our body. And we also have this delta carboxyglutamate. So delta, I think this is the attachment of the carboxylic group is found in the delta carbon. So this is found in the prothrombin. Prothrombin, this is responsible for blood clotting every time you wound yourself or you cut yourself. And then the second one, we have essential amino acid. Okay, now essential amino acid is being defined. These are standard amino acids needed for protein synthesis that must be obtained from the dietary sources. Now take note of this word. This must be obtained in dietary sources. Why? Simply because these amino acids are what they cannot be synthesized in our body. So since all of those other amino acids can be synthesized in our body, but these essential amino acids, we need them, but it just so happened that we cannot produce them. So we need to get these amino acids in our food. All right. And 9 out of 20 or 21 amino acids are considered as essential amino acids. So in these cases, we have here the names of essential amino acids in which it is not 9, but instead the one which is represented here are 10 essential amino acids. Why? Simply because, say we have here in the arginine, this is considered as essential amino acid for kids, responsible for their growth and development. But for adult, we don't or we no longer require arginine as our essential amino acids. We only need histidine down to valine. So we have histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. But for kids, they need arginine. So next we have the amino acids nomenclature and mnemonics. So nomenclature, this is how amino acids are being named. And I also included the mnemonics since I watched a video presentation, which is very nice, on how to easily grasp and remember. Yeah, the word is remember the amino acids, those 2021 standard amino acids, and how do we name them. So for the protein nomenclature, the common names assigned to amino acids are currently used, in which there are 3-letter abbreviations, which we usually see say you're going to search these 20 amino acids, they are written in 3-letter abbreviations, which is widely used for their naming. The first letter of the amino acid is compulsory and capitalized after, which is followed by the next 2 letters, which are not capitalized. So meaning to say, in this 3 letters, the first letter is capitalized and the succeeding second and third letters are no longer in caps lock. They are not capitalized, except in the case of asparagine, glutamine, and then we have tryptophan. Why they are in the exemptions? Because if you noticed here in my next slide, okay, so let's say for instance, we have this one, the arginine. Notice how their abbreviations, their nomenclatures are being followed systematically. So arginine, we have capital letter A, followed by R, followed by G. Histidine starts with letter H, followed by I, followed by S. Lysine start with letter L, followed by Y, followed by S. But notice how it is being done in asparagine, the glutamine, and tryptophan. They did not follow the 3 consecutive letters of the amino acids. Okay. Now here we have ASN. Supposedly that's ASP, but again there's an exceptions. For the glutamine, supposedly that's GLU, but that's GLN. And for the tryptophan, supposedly that should be TRY, but it is being written in TRP. Okay, remember that. Now, protein or the amino acids are also abbreviated in single word or 1-letter symbol, which is commonly used for comparing the amino acid sequences for the protein, especially in the sequence for us to easily remember them. So we use, most likely, we only use 1-letter word instead of 3-letter words. So usually the first letter of the name of the specific amino acids, and when there are more than one amino acid that has the same letter the most, so let's say um what the same first letter, so the most abundant amino acid gets the first letter, the rest they have different 1-letter symbol. Okay, so here we have arginine. So notice here for the alanine, we have alanine and arginine as well as the asparagine. They don't follow the letter A word simply because they are already in 4. We have here the asparagine. So only the alanine get the letter simply because that's what this is the most abundant. So therefore it it should be the one that is being abbreviated in 1-letter word according to how the spelling or their name started. The rest, they are in different letters. So for arginine, we have R, asparagine N, and aspartic acid for letter D. Same goes with the other amino acids. Now, what about for how they are, if amino acids are enchained already, how they are being named? So, in a peptide, amino acid is bonded to another amino acid through a peptide or an amide bond. Okay, so I was able to give justification already. I explained the difference between the peptide and the protein a while ago. Peptide, this is a shorter chain of amino acid, and protein, this is a chain of multiple peptides. If we're going to compare the size, peptide is smaller, protein is bigger. So by convention, the free amino acid or the N-terminal of the peptide chain is written to the left and the free carboxyl end or the C-terminal to the right. So here, say for instance, this is what this is already a combination, the aggregation of 3 amino acid, the glycine, the serine, and then we have the valine. Is it serine for the SER? Yeah, serine, I'm sorry. Glycine, serine, and the valine. So this is in the N-terminal and this is for the C-terminal. Okay. So therefore, all amino acids are read from N-terminal to C-terminal. So it always started from N to C, from left to right. Don't forget that. All amino acid residues have a suffix of in, ik, an, or ate, except for the tryptophan. Again, the tryptophan is one of the exemption, have a lot of exemption in terms of the nomenclature of the amino acids, so for which the suffix changed to YL or IL with the exemption of the C terminal. What does it mean? Those amino acids that ends with IAN, IC, IN, or ATE, except for the tryptophan, will be changed in YL and in N terminal, except that in C terminal, it will not change. You might find it confusing. No. Let's try this one as an example. We have the valine and the alanine. These are two amino acids joined together, okay, through hydrogen bonding. And then we have one, aside from this as a residue, we also have the water as the byproduct. So for valine, okay, we have this one, valine will change, IEN will change with YL, and then the C terminal, since this is the N terminal, the C terminal left untouched. Nothing happens to this one. So taken together, we have the valine and the alanine. We have the valine. Again, valine to valyl, then alanine stay intact. Taken together, we have the valylalanine. Example number 2. We have now 3 amino acids with aromatic ring. We have the threonine, we have the glycine, and then we have the tyrosine. Again, N terminal, C terminal starts from N, left untouched with the C terminal. Change into YL, threonyl, glycyl, and then tyrosine. Merge the name, we have threonyl glycyl tyrosine. Okay. Next, example number 3. We have now 4 amino acids attached into it. We have serine, we have phenylalanine, we have methionine, and then we have glycine. N terminal, C terminal, steryl, phenylalanyl, methionine, and glycine. Taken together, the 4 amino acids, we have seryl, phenylalanyl, methionine, glycine. Again, seryl, phenylalanyl, methionine, glycine. Okay. Next, we have 6 amino acid. I'm sorry. We have glutamate, we have lysine, we have cysteine, methionine, valine, and histidine. Again, N-terminal and C-terminal. Change into YL, glutamyl, lysyl, cysteine, methionine, valine, and histidine. Taken together, we have glutamyl, lysyl, cysteine, valine, histidine. Okay? So I hope you get already the concept on how to name the peptide. Now, I think I was able to mention earlier that amino acid have polar, non-polar, right? So for the polar amino acids, there are group or their side chains are polar. And basically there are 3 types. We have the neutral, acidic, and the basic. For the neutral, they contain polar but neutral side chains. For the acidic, they contain carboxyl group as part of their side chain. And for the basic, instead, this is the opposite. For the basic, they contain amino acid as part of their group in their side chain. In comparison, the opposite for the acidic, instead of amino, they contain carboxyl group. And here, you might be familiar with this one already. These are the 20 essential, or no, not essential, because essential includes already in the standard. These are the 20 standard amino acids, and all of these 20 amino acids are being classified according to their characteristics. I've shown you already the diagram of their classification. So number one, we have hydrophobic, or the water haters, or nonpolar, which is further divided into two classifications, either aliphatic, non-polar amino acids, meaning to say they have an alkyl groups in their side chains. We also have the aromatic, in which they do have an aromatic in their side chains. For hydrophilic, they are water loving. They love water so much because of the tendency to form a hydrogen bond with the water molecule for their amino acids. So they are divided into three classifications. We have basic, in which they are positively charged; acidic, which they are negatively charged; and polar or neutral, in which they are classified as uncharged. Now, other categories for amino acids can be alcoholic, amide, and then the sulfur-containing amino acids and essential amino acids. Now, based on, I watched already a video presentation about the mnemonics to easily remember the names of these 20 standard amino acids. So maybe you might find it helpful on your end as a student to easily memorize all of these amino acids. Now, for the hydrophobic aliphatic amino acids, since we have two classifications for hydrophobic, aliphatic and aromatic. For aliphatic, they have alkyl side chains, and we have this mnemonic of phobia of bump girl. Again, phobia of bump girl. What does it stands for? V stands for valine, A stands for alanine, M stands for methionine, and P stands for proline for the bump. For the girl, G stands for glycine, I stands for isoleucine, and no representation for the R, L for leucine. The second one, we have this hydrophobic aromatic amino acids. Aromatic meaning to say they have this aromatic side chains. And remember this mnemonic, aroma of pine and timber trees. Again, aroma of pine and timber trees. P stands for phenylalanine, T stands for tyrosine, and another T stands for the tryptophan. Now notice that I highlight the tyrosine amino acid here simply because tyrosine is one of the exemption for this hydrophobic amino acid, since they at some point they are also categorized as hydrophilic amino acid. Okay. And then we have now the hydrophilic, water-loving amino acids. We have this uncharged polar. Now, in uncharged polar, remember these mnemonics? We have come, take gas. Again, come, take gas. C stands for cysteine, T stands for threonine, G stands for glutamine, A stands for asparagine, and S stands for serine. Again, for hydrophilic amino acids, come, take gas. Next, we have these hydrophilic amino acids in which they are positively charged. Kanina, uncharged yun siya. Ngayon naman, positive and negative charge for hydrophilic. Now, for positively charged, remember the last action hero. Last action hero, which is very popular movie in the year 1993 that had a positive message. Now, L stands for lysine, A stands for arginine, and H stands for histidine. Again, for hydrophilic amino acid, positively charged. Last action hero, lysine, arginine, histidine. Now, what about for the negative charge or acidic hydrophilic amino acids? Now, remember this one, glue contains acid. Again, glue contains acid. G stands for glutamate, and A stands for aspartate. Now remember, for the negatively charged hydrophilic amino acids, these are the only amino acids that contains 8 at their suffix, the last portion of their name, 8, ATE, glutamate and aspartate. Next one, we have this alcoholic amino acids. So for alcoholic amino acids, remember these mnemonics. Alcohol tastes sour. Again, alcohol, this is a wrong spelling. Alcohol tastes sour. T stands for threonine and S stands for serine. For the amide amino acids, just the same as I presented in the negatively charged, we have the glutamate and the aspartate. In this case, we have G stands for the glutamine and acid stands for asparagine. Remember also that in this case, only two standard amino acids ends in INE, only the glutamine and the asparagine. For amino acids, amide amino acids, glue contains acid. Then for the sulfur-containing amino acids, meaning to say they contain sulfur in their structure, yes, the yellow one in the space filling model that I've shown you a while ago, remember the SCM. SCM stands for supply chain management. Again, SCM, supply chain management. S stands for sulfur, simply because they contain sulfur. C stands for cysteine, and M stands for methionine. Cysteine, methionine for sulfur-containing acid, SCM. Next, okay, so these are the points to note or to remember in the mnemonics and naming of nomenclatures. So all acidic and basic amino acids are charged amino acids, positive and negative charge. All hydrophobic amino acids are nonpolar. Hydrophobic meaning to say they are water-faring except for the tyrosine because this is an exemption. And all uncharged neutral hydrophilic and are what? Hydrophilic and tyrosine are polar. And then we also have this one, this essential amino acids. So we did mention already we have 10 for kids and 9 for adults as an essential amino acids, and we get them from the dietary food that we are eating or consuming. So we have two mnemonics for this one. We have private team hall. Again, private team hall. PBT for the abbreviation of private. P stands for phenylalanine, B stands for valine, and T stands for threonine. TEAM stands for tryptophan, isoleucine, and methionine. For the whole, H stands for histidine, A stands for arginine, L stands for leucine, and L stands for lysine. What about for mnemonic number 2? Okay, if you find it too hard to remember, then you could also have this one. These 10 valuable amino acids have long preserved life in men. These 10 valuable amino acids have long preserved life in men. Okay, so we have two T's here, threonine tryptophan, A for arginine, H for histidine, L for either leucine and lysine. P stands for phenylalanine, again for another L, lysine or leucine. I stands for isoleucine, and M stands for methionine. Okay, so if you find it so hard to remember, just go back with this video presentation. And so we're done already with the introductory for the protein structure and their level of organization. So in next video presentation, I will discuss the protein structure, primary, secondary, tertiary, and quaternary structure, as well as the functions. I will include now the functions in the second objective. See you